Okay, I think we are back in session, and we will ask Dr. Bocchini, who is already up at the podium, to get us started on the human papillomavirus vaccine session.
Thank you, John. Good morning. We're now going to begin a very important HPV vaccine session, and we'll describe that for you in the next couple of slides. Today we're going to have a presentation by Merck for a review of the investigational 9-valent HPV vaccine. As you know, this vaccine contains the 4 original serotypes in the quadrivalent vaccine and adds 5 additional oncogenic serotypes to the product. In addition, we're going to review and approve the updated HPV vaccine ACIP statement. Each of the committee members has reviewed the most recent draft, and we thank many of you for the comments that you've given us to improve the draft. This statement includes recommendations for the HPV 16 and 18 bivalent vaccine and the quadrivalent vaccine, and consolidates the information available for males and females. Just to review, in October, we provided an update on HPV vaccination coverage in the United States, and we heard from Merck on the 9-valent HPV vaccine development program. So you heard about the studies that were involved in the development of this vaccine. You also saw and contributed to a discussion on an overdraft of the overview of what was then the current draft of the updated ACIP statement. Since that time, the work group has had a number of conference calls. We've reviewed data from the 9-valent HPV vaccine clinical trials, and we've reviewed data on HPV type attribution and HPV-associated diseases. And we have now initiated discussions for grade consideration for possible licensure of the 9-valent HPV vaccine. So this is our proposed timeline for consideration of the 9-valent HPV vaccine. Today, and I'll flesh this out a little bit further, we're going to talk about attribution of types, HPV-associated disease, and you'll hear the first presentation of clinical trial data. In June, you'll hear additional information about clinical trial results and a health economics presentation. We anticipate in October to have the grade evaluation completed with recommendation options and discussion for ACIP. And feedback to make the final recommendations, which we expect to have in February of 2015, and pending the decision by FDA for licensure, a vote, and then an VFC vote as well. So today, the first 2 talks are on type attribution, HPV type attribution and cervical precancers by Susan Hariri from the CDC, and that'll be followed by HPV type attribution and HPV-associated cancers by Dr. Mona Soraya, who's also from the CDC. Our plan is to, and I'll talk a little bit more about type attribution in a second, but following these 2 talks to open this for questions for those 2 presentations. We'll follow that with a presentation by Dr. Elaine Luxemburg from Merck on the 9-valent HPV vaccine clinical trial data. And then Dr. Lori Markowitz will provide a summary and the next steps related to the 9-valent HPV vaccine program. And then she will review with us the updated statement and get additional feedback from the ACIP, and then follow that by an acceptance of that. So just to talk briefly about burden of disease, I mean, clearly as we advance vaccine development, it becomes very important to understand the role that various serotypes play in the development of the cancers and precancers associated with different serotypes of HPV. In addition, it's also important for us to establish guide for future evaluation of the impact of these vaccines. So the International Agency for Research on Cancer has defined some cancers as having a strong evidence of causal etiology. So for HPV-associated cancers, They include cervical, vaginal, vulvar, penile, anal, and oropharyngeal cancers, and the presence of HPV in these cancers is considered to be indication of causality. However, the determination of the type of HPV that's involved in a particular disease really depends on a number of factors, which include the quality of the specimen that's being evaluated, the assay used to detect the HPV, the algorithm to assign attributable type when multiple types are discovered in the same preparation, and they can vary based on a population sampled. And that's why it's important for us to have these two presentations, which will give us some understanding of where we are and the programs that are available for us to get a better picture of the relative roles of various serotypes in precancer and cancer lesions. I want to thank this work group. The members of this work group have been very active and involved in our conversations on the phone and in other efforts. Particularly want to thank Lori Markowitz for her organizational skills and her ability to keep this committee on task, and thank the ACIP members for their contributions as well. So with that, I'd like to turn this over to Dr. Herrera, who will provide the first presentation. Thank you. I forgot to mention one thing. The ACIP members have a full copy of Dr. Luxemburg's presentation. However, what we found is that for the public and those around outside tables, only half of the talk has been copied. You're missing every other page. And so we apologize for that. And so the plan is to make sure that you're all aware that these slides will be published. They'll be put on the web when the slides become available. So thank you.
Good morning. Today I'm going to be presenting data on HPV type attribution in high-grade cervical lesions in the U.S. Oops. Okay. I'm going to start with briefly reviewing the natural history of cervical HPV infection and associated outcomes. And then I'll review some published data on the distribution of HPV types in high-grade cervical lesions in women from the different areas of the world. And I'll also review methods used to estimate individual HPV type attribution and the reasons for them. And then finally, I'm going to present data from the US on HPV type attribution to non-invasive high-grade cervical lesions. As you know, infection with genital HPV is responsible for a spectrum of cervical abnormalities. The majority of these abnormalities are benign and don't progress to disease. In fact, about 90% of infections become undetectable within 2 years without causing any symptoms. However, infection with high-risk HPV types can persist, and that could lead to mild cervical abnormalities that tend to regress without treatment, or to high-grade lesions that are more likely to progress to invasive cervical cancer if they're not treated. And routine cervical cancer screening with the PAP and more recently with HPV-based tests is recommended to detect and treat precancerous lesions as a secondary cervical cancer prevention strategy. So the focus of this presentation is on non-invasive high-grade cervical lesions, specifically on cervical intraepithelial neoplasia grades 2 and 3 and adenocarcinoma in situ, which I will be collectively referring to as CIN2+. CIN2+ lesions are associated with high-risk or oncogenic HPV types that are the most likely to persist of all the HPV types. About 30% of CIN2+ lesions are estimated to progress to cervical cancer. HPV-16 is the most common type in CIN lesions, which are squamous lesions, whereas HPV-18 is the most common type in AIS lesions, which are glandular. CIN2+ lesions were used as the main endpoints in all the vaccine efficacy trials, and they're also used as intermediate endpoints to monitor post-licensure vaccine impact in some countries that have established screening programs. So this graph was adapted from a meta-analysis of international studies that were conducted to determine HPV type distribution in non-invasive high-grade cervical lesions in women around the world. And the high-risk HPV types here are combined into two groups. The blue bars represent HPV type 16 and 18, and the red bars include HPV types 31, 33, 45, 52, and 58, which are the 5 additional types in the candidate 9-valent vaccine. And so you can see from the blue bars that there's variation globally in the proportion of HPV 16 and 18 detected in high-grade cervical lesions from over 60% in women in North America, Europe, and South and Central America to less than 50% in women in East Asia and in Africa. There's less variability in the 5 additional types, with about 40% detected in women across all regions except for in East Asia, where it was detected in 60% of lesions. So the objective of this analysis was to evaluate high-grade HPV type attribution to cervical intraepithelial neoplasia and adenocarcinoma in situ among U.S. women aged 21 to 39 who were diagnosed from 2008 through 2011. And we also wanted to examine whether HPV type attribution differed by race and ethnicity. So these data come from the HPV Impact Project, which is a population-based sentinel surveillance system that was established in 2008 to monitor HPV vaccine impact on high-grade cervical lesions and HPV types associated with them. This is a CDC collaboration with 5 emerging infections program sites, and it includes catchments of similar sizes with about 300,000 women in each catchment in California, Connecticut, New York, Oregon, and Tennessee. And at each site, histopathology laboratories are enumerated and asked to report histologically confirmed CIN2+ diagnoses in adult residents of the catchment who are 18 and older. Labs are also asked to submit archived diagnostic tissue from women who were diagnosed before the age of 40 for HPV DNA typing. Depending on the number of women reported and resources available, the sites request all or a random sample of specimens from age-eligible women from each reporting lab. And labs are asked to select tissue that's most representative of the diagnostic lesion and to cut and process the tissue according to standard protocol for formalin-fixed, paraffin-embedded specimens. And then the processed specimens are sent to CDC, and here they're reviewed again by a pathologist to ensure that the tissue is adequate for typing. And if it is, then DNA is extracted and tested for 37 individual HPV types using the Roche linear array genotyping assay. And for specimens that are negative on linear array or fail to amplify control, they're retested using a second genotyping assay, and that represents about 10% of the specimens overall. So between 2008 and 2011, we had adequate typing results for over 5,000 specimens, and almost all of them, or 97%, were positive for HPV, and only a single type was detected in 79% of the specimens. HPV 16 and 18 was detected in a little over half, or 53% of all specimens, and HPV 31, 33, 45, 52, and 58 were detected in 31% of the specimens overall. So these are typing results broken out by severity of lesion into CIN2, which is the lowest histologic grade, or CIN3 and AIS, which are the highest grade lesions. And AIS here is combined with CIN3 because of small numbers, because it represents less than 2% of all the reported cases. And there's also about 900 specimens in which specific histologic grade was not discriminated, and they're not included here in this table, but they do represent a combination of the two grades. And some things to note here is that CIN2 accounts for a larger fraction of all CIN2+ diagnoses, and that 16 and 18 types are more common in CIN3 and AIS compared to CIN2, 66% versus 40%, whereas the other 5 additional types in the candidate vaccine are actually more common in CIN2 compared to CIN3 and AIS. This graph shows the prevalence of individual high-risk HPV types by histologic grade. And just to orient you, the blue bars, I don't know if I have the blue bars here indicate CIN2 lesions. The red bars are those that are not discriminated and are labeled as CIN2/3, and the green bars are CIN3 or AIS. And then the HPV types are grouped along the x-axis. And so the first group are HPV 16 and 18. And what's notable here is that 16 is detected by far the most detected type in all of the lesions, although it does increase from 35% in CIN2 lesions to 60% in CIN3 and AIS. And HPV 18 is relatively uncommon and accounts for about 5 to 6% of lesions. Then the next set of types are those in the candidate 9-valent vaccine, and here HPV type 31 is the most common, followed by HPV types 52 and 58, which are both more common in CIN2 than CIN3. And then 33 and 45 were detected in less than 5% of specimens across all histologic grades. The last set are other oncogenic or high-risk types that are not included in any vaccine. And here you can see that types 31 and 55 are the most common, and that the other types occur in 5% or less of specimens across all histologic grades. So for the purpose of HPV type attribution to cervical lesions, usually when only a single HPV type is detected in a lesion, that type is considered to be causally associated with the lesion. However, more than one HPV type is detected in about 20% of CIN2 plus lesions, and the role of each type in these co-infected lesions is less clear. And so different methods are used to attribute individual HPV type or group of types to cervical lesions, and these methods are used to evaluate the potential impact of HPV vaccine. So in this analysis, we applied the 2 most common methods that are used for HPV type attribution. The first is hierarchical, which basically assigns attribution to the most oncogenic type in a lesion. So, and those are usually 16 or 18. So just to give you an example, if you have a specimen, a CIN3 specimen with HPV 16 and 31 detected in the lesion, the whole specimen would be attributed to HPV 16. So this tends to overestimate HPV type 16 and 18 because they're the most oncogenic types of all the high-risk types. The second method is a proportional attribution method, and here weights are assigned proportional to the type frequency in single infections in each disease grade. And so to use the same example, we would calculate the proportion of each type that occur as a single infection in that histologic grade. And so here it would be 61% of HPV 16 and 9% for HPV 31. And then that is divided by the sum of the total attribution of all the types in the lesion. And we found that there was a less than 2% difference between the 2 methods that we applied, and so the results that I'm going to be presenting are based on the proportional attribution method. Okay, so here is the proportional type attribution by histologic grade. The blue bars again are CIN2, the red bars are CIN2/3, and the green bars are CIN3 or AIS. The lighter portion of each bar represents lesions with single type infection, and the dark part at the top is multiple infections. And as you can see, 38% of CIN2 lesions were attributable to HPV 16 and 18 types, and the percent attribution of 16 and 18 increased with severity of lesion up to 64% in CIN3 and AIS. 28% of CIN2 and slightly less of higher grades were attributable to the 5 additional types in the candidate vaccine, and the other high-risk type attribution ranged from 21% in CIN2 to less than 10% in CIN3 and AIS. So this graph shows you the proportional HPV type attribution by age group. And here the bars represent the groups of HPV types. The blue bars are 16 and 18. The red bars are the 5 additional types, and the green bars are the other high-risk types. And as you can see, HPV type 16 and 18 are attributable to the highest proportion of lesions across all age groups. However, there is a decline in the proportion of HPV 16 and 18 in the oldest age group. And in that group, we see an increase in the 5 additional types. And as far as the other high-risk type attribution, it's fairly similar across all age categories. This graph is similar to the previous one, except that this is proportional type attribution, this time by race and ethnicity. Again, I want to bring your attention to the fact that HPV 16 and 18 are attributable to the highest proportion of lesions across all racial and ethnic groups. However, HPV 16 and 18 attribution is higher among non-Hispanic whites compared to the other race and ethnic groups. Conversely, the 5 additional types are more common in racial and ethnic minorities compared to non-Hispanic whites. And the other high-risk types are significantly higher in non-Hispanic blacks compared to the other racial and ethnic groups. So the previous slide showed you the type attribution by groups of types, and here you can see the high-risk HPV type attribution broken out by individual types, again by race and ethnicity. Again, I want to point out that HPV 16 is the predominant type across all racial and ethnic groups. It is higher in non-Hispanic whites compared to the non-Hispanic blacks and Hispanics. HPV 45, 52, and 58 were significantly different by race and ethnicity among those that are in the candidate 9-valent vaccine. And then the last 2 types on this graph are those that occurred in at least 5% of lesions in at least one of the racial and ethnic groups. And you can see that HPV 35 here is significantly higher in non-Hispanic blacks compared to the other 2 groups. So in summary, these data from a large population-based sample of women in the U.S. showed that 50% of CIN2 plus lesions are attributable to HPV 16 and 18, and the attribution ranges from 40% in CIN2 lesions to over 60% in CIN3 and AIS. Another 25% of CIN2 plus lesions are attributable to HPV 31, 33, 45, 52, and 58, which are included in the candidate 9-valent vaccine. We found that there was a higher proportion of lesions due to HPV 16 and 18 in women who were under the age of 35, and this is consistent with evidence that indicates HPV 16 and 18 are stronger carcinogens and more likely to progress faster to disease. We also found that the highest proportion of CIN2 lesions were attributable to HPV 16 and 18, irrespective of racial and ethnic category. However, there was a higher proportion of lesions attributable to 16 and 18 in non-Hispanic white women compared to other racial and ethnic groups. And the reasons for these differences are not clear and are probably multifactorial, but may be due to differences in the underlying prevalence of HPV infection in the different subpopulations or to differences in screening and treatment. Thank you.
Thank you much. We're going to hold a comment and question until after the next presentation by Dr. Soraya.
Good morning. What did I do? What? The other way? Okay. Good morning. Before I start talking about attribution, I wanted to review these data from the annual report to the nation published last year, showing the incidence rate of cervical, vaginal, vulvar cancers. The orange bar represents all race ethnicity, and the teal bar Hispanic, and the purple bar Black, and so on. And what I wanted to show was that the Black women and Hispanic women have the highest rate of cervical cancer, and Black women also have the highest rate of vaginal cancer. The rate of oropharyngeal and anal cancers by race are seen here. And here what I want to point out is that Black males and females, as noted by the purple bar, have the second highest rate of oropharyngeal cancer, and for anal cancer, Black males have the highest rate. These are international data of archived cervical cancer specimens, invasive cervical cancer specimens, collected from all around the world and typed by one lab to determine what percentage have 16/18 and the additional types, 5 types in the candidate 9-valent HPV vaccine. And the title is called Relative Contribution because the denominator data is among HPV positive cancers. And when the denominator data is among all cancers, the terminology used is attribution. So important issues to highlight here are that the majority of cancers, whether they're in Africa, Asia, or Europe, have 16/18, usually in the range of around 70%, and an additional 17 to 20% have contribution by the other 5 types. The other thing I wanted to point out here was that there have been very few cases that have been analyzed in North America, around 160, from this comparative study, and that's why we undertook the CDC study I'll describe later. The same group that did this particular study has also conducted relative contribution for other cancers, but most of those data are not available. This slide summarizes literature from US studies prior to 2006 that have examined tissues from these particular cancers and attributed them to HPV. So the first column summarizes the attribution to any HPV, and the second column summarizes attribution to 16 and 18. So for cervical, for example, you see 96% was attributable to HPV, and among all cervical cancers, 76% were attributable to 16-18. And for vaginal, 64% for any HPV and 56% to 16-18. The objectives of this particular analysis were to establish systematic population-based approach to monitoring the HPV types and cervical as well as other HPV-associated cancers, and to determine the attribution of 16-18 and additional types in the candidate 9-valent vaccine, and also to see if there were differences by race ethnicity. So the study design, basically we used cancer cases that were identified through cancer registries, most of which were collected from 2004 to 5, right before the vaccine was licensed, to help us give a baseline. The registries have information about the cancer diagnosis, but in order to get the actual tissue specimen, 4 of the cancer registries went to pathology labs to get the most representative specimen, and 3 had existing biorepositories. Most of the cancers that were collected were invasive cancers, but we did have a few in situ cancers like cervical in situ, which is equivalent to CIN3, because the registries had the legal authority to collect these data. The HPV genotyping was done by the CDC lab, as described by Dr. Susan Harari, and the attribution method was the same. The way that we categorized HPV was HPV 16-18, the additional 5 types in the nanovalent vaccine, any other HPV types, and HPV negative. And the denominator included all cancers. This figure is a schematic of the tissue sample request and submission process. So on the left-hand side, the 4 cancer registries, Michigan, Louisiana, Florida, Kentucky, generated a list of all eligible cancer cases or precancer cases, sampled them, and then requested the local hospital or pathology lab with the tissue specimen to prepare and send to the CDC HPV lab. On the right-hand side, what you see is Hawaii, Iowa, and Los Angeles County. What they did was they already had existing tissue repositories, so they prepared the samples and the tissue in-house and sent them directly to the CDC HPV lab. And in the path labs, the tissue specimens were prepared according to a common standardized protocol And then the complete genotyping results were then linked with the demographic data collected by each of the cancer registries. And so we had 3,017 tissue specimens that made it to the CDC HPV lab, and due to either specimen inadequacy or other issues, we had a final sample of 2,670 cancers. This slide shows the 8% HPV detection by cancer site. So in the study, you also see the number of cancer cases for each of the sites. 91% had HPV of cervical cancers had HPV DNA detected, 99% of in situ cervical cancer, 69% of vulvar cancers had HPV detected, 75% for vaginal cancers, 91% for anal, 63% for penile, and 70% for oropharyngeal. This slide shows the top 5 cancer-causing types in these select cancers. And the slide may be a little difficult to see in terms of the specific types, but the big broad picture is that HPV-16 is the most common type in all of these cancers. And I will go over these cancers in a little bit more detail. Here we have attribution by the 4 HPV groups, blue being 16-18, and yellow the additional 5 types in the candidate vaccine, green being other HPV types, and gray HPV negative. So for cervical cancer, you see that 66% of the cancers had 16-18, attributed to 16-18, additional 15% attributed to the candidate, or the 5 types in the candidate vaccine, 10% for other types, and 9% were negative. So anal cancer had the highest proportion of 16-18 detected, and for the additional 5 types in the candidate vaccine, the cancers that had the highest attribution were in situ cervical cancers, vaginal, and vulvar cancers. So we conducted lots of analyses, and the general findings, which we'll go into more detail, were that by age, there was definitely a higher proportion of cancers in younger age attributable to 16-18. By race ethnicity, there were no differences for cancers except for the in situ cervical cancer and the oropharyngeal cancers. And by gender, there were no differences for the cancers except for oropharyngeal cancer. This slide looks at the attribution by race ethnicity for invasive cervical cancer on top and in situ cervical cancer on the bottom. And there are racial differences that are noted in terms of a lower proportion of 16-18. Lower proportion of 16-18 for among black non-Hispanic populations, as well as a lower proportion among Hispanic populations. So this pattern is similar to what was seen by Hariri, but we have to keep in mind that for in situ cervical cancers, we did have a lower number of samples. However, when you examine the invasive cancers on top, and we have a pretty good sample size here, what you see is that HPV negative cancer proportion was lower among blacks and Hispanics than whites, but more importantly, there were no differences in the proportion of 16 and 18. 67%, 68%, and 64% by the different racial ethnic groups. Many of you might ask, how can cervical cancers be negative? We have this previous study that has found 99.7% to be HPV positive, but we have to keep in mind that the study was based on multiple HPV assays that were done and cancers that met select criteria, and what we are doing is a population-based surveillance study. There may be misclassification of the actual anatomic site. In fact, in that the lower segment uterine cancers cannot be distinguished from the upper segment cervical cancers. There may be false negatives in that there is HPV, but we can't detect it with the current assay, or the specimen and tissue is not well preserved. And then there are actually true HPV negative cervical cancers, albeit that they're rare histologies, maybe 1% to 2% at the most. This slide looks at HPV attribution by race and gender for anal cancer. And here on the left-hand side, what you see is that there's not many differences by race ethnicity. For 16-18 attribution, around 80% are attributed to the 16-18. And also by gender, on the right-hand side, there are no differences, 79% to 16-18 compared to 80% among females. Although it appears that females may have a higher percentage of the additional 5 types in the candidate 9-valent vaccine, this difference was not significant. This is oropharyngeal cancer, an HPV attribution, and on the left-hand side, what we see is that the race for HPV 16-18, what we see is that there's a higher proportion of HPV-negative cancers among blacks, around 50% compared to 25 to 27% among whites and Hispanics, and a lower proportion of 16-18 cancers overall among blacks, even when the cancers are limited to HPV-positive cancers. By gender, there's also a higher proportion of HPV-negative cancers among females in gray, and a lower proportion of HPV 16-18 cancers as well. So if we use the data from this particular study and revised our estimated percentages, we can definitely say with more authority using population-based assays, consistent assays, that our attribution of HPV positivity would be slightly revised. For cervical, we see 91% of cervical cancers would have any HPV attributable. 66% of all the cervical cancers would have 16-18 attributable, and 15% for the additional 5 types in the 9-valent vaccine. And on the same note, like for example for oropharyngeal, for males, 72% would be HPV attributable, much higher than our previous estimates, and 63% have 16-18, and 4% have the additional 5 types in the 9-valent vaccine. So in summary, 62% of invasive cancers are attributable to 16-18, and this ranges between 48% of penile cancers to 79% of anal cancers. And when you look at it by gender, 62% of females are attributable to 16-18 compared to 63% for males. And this translates overall to approximately 25,000 cases annually. 11% of the invasive cancers are attributable to the additional 5 types in the candidate 9-valent vaccine. And this ranges based on the cancer type, 6% for oropharyngeal and 18% for vaginal. It also differs by gender, 14% for females compared to 5% for males. And overall, this translates to 4,000 cases annually. For racial ethnic differences, the attribution for 16-18 did not differ for invasive cancers except for oropharyngeal cancers, in that there was a lower percentage of oropharyngeal cancers attributable to HPV positive or HPV 16-18 among blacks. These data will be useful in estimating the impact of the candidate 9-valent vaccine on cancers, as well as for cost-effective analyses. I'd like to acknowledge the co-authors for this particular study, as well as this presentation.
Thank you. I think we're open for questions. Dr. Engold.
Very helpful presentations. Susan, I think you know I'm a big fan of the impact studies, but I wonder if I understand correctly, the cases that you're looking at here, they span a time range before and after when the vaccine was being used. And given what we know about coverage, which was obviously fairly low, given what we know about latency, I'm just wondering whether we might be underestimating the amount we can attribute to 16 and 18 if we're including in the studies some of the cases occurring basically after the vaccine is already being utilized, or if you think that's an issue or not.
Sure, that's quite possible. Thank you for that comment. As you know, we within the HPV Impact Project, one of the things that we try to do is collect HPV vaccination history on all the cases, and we sometimes can't find HPV vaccination history on everybody, but we do have vaccination history on about 50% of the cases, and among those that we had vaccination history for, there was a pretty sizable proportion in the 18 to 20-year-old females with CIN2+. And I did not present that data here. I actually excluded those. There were a few in the 21 to 29-year-old group, but when we excluded those from the analysis, it didn't significantly change the results. So it's possible that there is some impact, but because we don't have, vaccination history on 50% of the cases, it's really hard to tease that apart.
Dr.
Yes.
Curran.
So thank you very much for those presentations. I wonder if you could remind us about cross-protection against non-1618 serotypes from the vaccines. Which serotypes might there be protection against, and do the vaccines differ in their ability to do that?
So there has been some cross-protection shown for both vaccines, mostly in types 31, 33, and 45. And I think that the cross-protection has been a little bit stronger for the bivalent vaccine, although it's really difficult to compare the two vaccines head-to-head. So yes, there is some cross-protection that's been demonstrated. The duration of protection is unknown. But yeah, there does seem to be some cross-protection.
Ms. Hayes.
Carol Hayes with ANA and ACNM. Dr. Harari, I had a question related to your slide 11 and 17. And you may not be able to answer the question, but when I looked at the data, it appeared to me that there were two strains of HPV that seem to be higher than the ones that are used in the new 9-valent vaccine. And I'm confused by that. The ones over on the right, you can see that 51 and 35 are higher than 33 and 45. So why were 35 and 51 not included in the vaccine, and why was 33 and 45?
I'm not sure that I can really answer that question. I mean, one thing that I want to point out, though, is that the prevalence is very low compared to type 16 across the board for all of the types. And you're right that 35 and 51 are more common, but that's mostly in the CIN2 lesions, which are kind of a lower histologic grade and are probably not the true precancers. The prevalence is significantly lower in the CIN3 and AIS lesions. But as far as, the decision to which types to include in the vaccine, I can't really speak to that.
Yeah, and just for, these are the precancers, and most of the decisions were made based on the types and the cancers. And many of these, even these are oncogenic types, they're less likely to progress to cancers than 1618.
And I believe Dr. Cooter has a comment back at the microphone on that.
Yes, what Lori said is correct. The decision that we made in terms of what additional types would go into the vaccine was actually based on our global data, looking at the prevalence of all of these types around the world. It was based primarily on oncogenic cancer types. So that's why 31, 33, 45, 52, and 58 are in the current vaccine. So there is a little bit of a difference in the epidemiology between the U.S. data shown here and the global rates.
Thank you. Dr. Bennett.
Thank you. And thanks for this great presentation. It's really terrific. I'm very interested in the racial differences in Susan and your data. And I was curious if you could remind us what was shown in the carriage data, which from the NHANES survey. Were there racial differences in those data as well?
So we can actually monitor HPV prevalence using the National Health and Nutrition Examination Survey, which is a national survey of the non-institutionalized U.S. population. And we've looked at HPV type prevalence in NHANES over time. And yes, Dr. Bennett, it's true that there are racial differences, but mostly the differences are that there's a higher prevalence of types, of all types, in non-Hispanic blacks compared to non-Hispanic whites. And we're sort of we're in the process of looking at the data more closely, but there doesn't seem to be a differential increase or decrease in the prevalence of the oncogenic types. 16 and 18. So it's just higher for all the types as far as we can tell, but we're looking at that data now.
Can I ask a follow-up question? So do you have any thoughts about why we would find that pattern in the early lesions and not invasive cancers? And not in, and it sounds like not in the NHANES data either, that differential.
Differences in 16 and 18? So as I said, 16 and 18 are more oncogenic and have more oncogenic potential than the other oncogenic types. And as you saw in Dr. Soraya's presentation, we don't really see any differences by race when we look at the cancers, the invasive lesions. Like I say, it's really, we're not really sure what to attribute the differences, the racial differences to, but it could have something to do with the prevalence combined with the differential oncogenic potential of the types. But again, that's something that needs to be further investigated.
Any other comment or question? All right. Thank you. I guess we move along to the presentation from Dr. Luxembourg.
Okay, good morning, everyone. And we are going to now talk about the clinical program for the 9-valent vaccine. This is the second presentation at ACIP. We presented already in October of last year the program design. And this time we are going to show you some of the key data from the clinical program. Doesn't seem to work. Okay, you will pass the slide for me. Just a few abbreviations for everyone to be on the same page here. So we will talk about two HPV vaccine. One is the 9-valent vaccine, which is the investigational vaccine, and it's abbreviated 9V-HPV. And the licensed vaccine, Gardasil, the licensed 4-valent vaccine is quadrivalent, so QHPV. Genital lesions that we'll talk about have been also mentioned in the previous talk. CIN, cervical, intraepithelial neoplasia. VIN, vulvar, intraepithelial neoplasia. Vein, vaginal intraepithelial neoplasia. And as mentioned in the previous talk, grade 1 is a low-grade lesion. It's a reflection of persistent infection. Grade 2, 3 is a high-grade lesion. It is a precancer. Just a few slides of recapitulating epidemiology worldwide, and you will see it's very consistent with what has just been shown. This is a relative contribution of HPV type worldwide. The existing vaccine covers type 16 and 18, which represents 70% of cervical cancers worldwide. By adding the 5 next most frequent types in cervical cancer, we have the potential to reach 90% prevention. And so to answer the question that was asked previously, the choice of the 5 new types was based on the frequency in cancer, not in precancer and dysplasia, in cancer worldwide. And the 5 types that are also oncogenic that are not included represent 5 to 6% cervical cancer worldwide. Looking now at precancers, again worldwide, there is also a potential for additional coverage with the new types, and especially looking at SIN2-3, the precancers, there is a potential to increase protection from 50% with the existing vaccine to 80%, which exceeds the best cervical cancer screening programs. So it's an exciting possibility. As you can see, the contribution in cancer is slightly lower than in precancer, and most likely due to a faster progression to cancer with type 16 and 18, as was discussed before. So the potential, in the previous slide also, the potential is not only prevention of cancer worldwide, but it's also potential of prevention of precancerous lesion, which means in countries with cervical cancer screening program, lower need for invasive procedures, because these precancers are treated by surgery. Lower cost, lower healthcare utilization, better quality of life, lower risks. So there are really a wide range of benefits that could be anticipated from the 9-valent. Here on this slide, you see the 4-valent vaccine. It's a VLP-based vaccine. The VLP are the virus-like particles. They are like the capsid of the virus. It looks like the virus, but it doesn't have the DNA in it. Outside, it looks like the virus. It's seen by the immune system as the virus. So the 4-valent contains 4 types, 6 and 11, which are responsible for 90% of the genital warts. They are generally responsible for benign lesions. And type 16 and 18 responsible for most cervical cancers. The 9-valent contains the same 4 types plus the 5 new types. The adjuvant is the same. It's an aluminum-based adjuvant. The amount of adjuvant has been increased to keep the adjuvant to antigen ratio the same. And the amount of antigen for the original types has also been increased to preserve immunogenicity. So all these were data-driven decisions. The goal is to transfer from 4-valent to 9-valent, contains the same types, and the clinical program was designed to assess that. And the goals of the clinical program are to show that for the 4 original types, the 2 vaccines perform the same. We want to transition fully. We want the second vaccine to do the same as the original vaccine. In addition, for the new types, for the additional types, we'd like to demonstrate that the new vaccine provides substantial benefit, which means is highly protective against infection and disease due to the additional types. Adolescents are an important target population. However, we cannot assess efficacy in adolescents because they are not exposed to HPV most often. And so similar to what was done in the 4-valent program, the efficacy findings in young women exposed to HPV will be extended to adolescents based on similar immunogenicity. And so the program is designed also to demonstrate that. And finally, obviously, we want to demonstrate that the safety profile is acceptable. So really 4 goals. If this can be demonstrated, it would show in our mind value for the new vaccine. And this is done with 6 phase 3 studies, which are included in the initial filing. On this slide, you have the 3 pivotal studies that support the key indication, we hope. And the study results will be presented today. Protocol 1 is the only efficacy study. It's 2/3 of the program. It's in young women. Study is completed. And an extension, long-term extension is ongoing to assess long-term effectiveness and long-term immunogenicity. In adolescent, as I mentioned, we conducted immunobridging study. Protocol 2, an adult to adolescent immunobridging from young women to adolescent. The base study is completed and an extension is ongoing now for 10 years also to assess long-term effectiveness and immunogenicity. And finally, protocol 9 is to assess whether the 4-valent and the 9-valent induce the same immunogenicity in adolescents. So these are the pivotal studies. Next slide is going to show the supportive studies. So 3 more studies that will be also included in the initial filing and will be presented at a next ACIP meeting. Concomitant use study assessing assessing vaccines that are commonly used in adolescents and co-administered with the 9-valent. Protocol 5 addresses Menactra, a meningococcal vaccine, and Adacel, Tdap vaccine, commonly used in the Americas. And Protocol 7 addresses Repevax, a Tdap polio vaccine commonly used in the European Union. The 2 studies are completed. And finally, there is one study that's unique to this program, which is to assess the vaccine, the 9-valent vaccine in prior Gardasil recipients. We recognize that there has been millions of individuals vaccinated with Gardasil, and among them, even if a small fraction is interested in a follow-up vaccination with 9-valent, we feel obliged to provide data so that providers and individuals can make their choices. We are not looking for an indication in that case, but we feel it's going to be an important study to discuss. And finally, you could ask, where are the young men, because the initial filing is in boys 9 to 15 years of age, girls and young women 9 to 26 years of age. The young men study is ongoing. It started a little later, but we'll have the study the result later this year. These are not the only studies ongoing for the 9-valent program. For the sake of time, we cannot present everything, but I'd like to mention also one study that may be of interest for this group. We have recently started an evaluation of a 2-dose regimen for V503, for the 9-valent vaccine. Why the 9-valent vaccine? It's because now the focus is on the 9-valent and not on the 4-valent. We expect a full transition very rapidly in the US, and so the focus for the 2-dose is on 9-valent. We have an initial study started to address the question of feasibility and value, and we recognize also that long-term effectiveness is going to be needed as well. So today we will talk about study results data. And the data will focus on evaluation in young women, efficacy and immunogenicity and safety, data from Protocol 1, the pivotal efficacy study, and then we'll turn to evaluation in adolescent. It's going to be immunogenicity and safety, Protocol 2 and Protocol 9, the 2 immunobridging studies in adolescent. Protocol 1 is a very large study. It enrolled more than 14,000 young women. It's a double-blinded study. Women were randomized to 9-valent or 4-valent. We didn't use placebo for ethical reasons. The 4-valent vaccine is widely recommended, is widely available, and we cannot let people develop precancer in a study. So the vaccines were administered as a 3-dose regimen. Keep in mind that the study was started in 2007. It was started 1 year after Gardasil was licensed. So it was really looking forward. And at that time, the focus was really to bridge from Gardasil to the 9-valent. The key endpoints are efficacy. Women are followed by gynecological examinations. There are genital swabs to assess for the presence of HPV, as well as Pap test every 6 months. And for any subject with an abnormal Pap test, there is a protocol-mandated triage to assess for disease endpoints. Immunogenicity is also assessed, primarily at day 1 and month 7 for titers for all anti-HPV types. And finally, safety also assessed through standard methods. So it's once again, it's a very large effort, it's a very large trial. It's been going on for almost 6 years now. And subjects have had a follow-up up to 4 years post-vaccination. There was an intensive screening every 6 months to make sure we don't miss any lesions. So we have a very high quality data package here. Next slide is showing the primary objective. So once again, we don't have a placebo control. Our control is the existing 4-valent vaccine, which we all know is highly efficacious. Essentially, we expect no disease endpoints for the original types. And if the 9-valent is successful, we expect also no disease endpoint or very few for the 9-valent for the original types, which means that a head-to-head comparison based on efficacy endpoints is not feasible. We will have too few endpoints to make a valid statistical comparison. So based on that, the demonstration of efficacy for the original types, the primary demonstration of efficacy is based on non-inferior immunogenicity. It's an immunobridging, the goal being to bridge the efficacy findings with the 4-valent vaccine to the 9-valent vaccine based on non-inferior immunogenicity. For the additional types, we expect a difference between the two vaccines in terms of disease endpoints. As mentioned, there is some cross-reactivity and some cross-protection, but it is still, insufficient to cover all the types. And so for the analysis, I will show you the 4-valent cohort will be assimilated to an unvaccinated cohort for the additional types. If anything, it would mean an underestimation of the true efficacy. So here we have really two different primary objectives for the original types and for the additional types, and we'll show the data in a moment. The population that are used for the primary analysis are susceptible population. We want to analyze a prophylactic vaccine in women that are not infected to the relevant type and follow them during the study for infection and incidence of infection, incidence of disease. So similar, the definitions are very similar to the one used previously in the 4-valent clinical program. Subjects have to be negative at baseline for HPV and throughout the vaccination period. And, obviously they need to not be a protocol violator and receiving 3 vaccinations. And we have also for immunogenicity a per protocol definition as well. Subjects received 3 doses. Most of them stayed in the study, so the compliance with the study was very good, and there was no difference between the two vaccine cohorts, and over 96% continued in the efficacy evaluation phase. We are looking here at, you remember we are looking at original types first, and we are looking at immunobridging, non-inferior immunogenicity. We are looking here at month 7 antibody titers measured by CLIA, which is a competitive Luminex immunoassay used by Merck, also used previously in the 4-valent program. And what we demonstrate is non-inferiority for all 4 types, with GMT ratios that are generally close to 1. And, really, this demonstrates that the primary efficacy is fulfilled here. Seroconversion rates were also comparable between the two groups, with virtually every individual vaccinated with a 3-dose regimen becoming seropositive after 3 doses. Let's now examine the data for the additional types. And that's our second primary objective. Here we are looking at 3 efficacy endpoints. These are composite endpoints looking at disease or infection caused by any of the 5 new types. So on the first row, it's our primary endpoint, high-grade cervical, vulvar, and vaginal disease due to any of the new types. Then we have cervical, vulvar, and vaginal disease of any grade due to any of the additional types, and then 6 months % infection. And what's remarkable is that the efficacy is very high, over 96%, very consistent across all endpoints. And so, it gives a very strong argument that the vaccine is highly efficacious in reducing the risk of disease and infection due to the new types. In addition, these very high numbers for efficacy are reminiscent of the numbers for efficacy that were found in the 4-valent program, where also the efficacies were in the high 90s. So we have now 2 vaccines that are based on the same technology, based on VLP, and in 2 different clinical trials showed to be highly efficacious with efficacies that are in the high 90%. So we have really 2 vaccines from the same family. You have a family of vaccine here, a family of product. Next slide. Here we are looking at cervical and vulvar vaginal disease taken separately. And this slide shows you that for high-grade cervical, all-grade cervical disease, high-grade vulvar and vaginal disease, all-grade vulvar and vaginal disease, efficacy was very high as well. And I'd like to mention that we have one case of high-grade cervical disease in the 9-valent group. And this case is co-infected with a non-vaccine type, with type 56. And most likely type 56 is a causal virus because this subject was infected by type 56 from day one and through the first 2 years, whereas type 58 was found only at the time the biopsy was done. The type 58 was most likely a transient infection. Next slide. Next slide shows invasive procedures that are associated to the additional types. And you can see here that biopsies and definitive therapies caused by the discovery of the lesion, the risk of this invasive procedure is decreased in a very substantial manner as well, which tells you that not only the vaccine has the potential to prevent a lot of disease to the new type, but it has the potential also to prevent a lot of procedures. Let's go now to safety. Vaccine-related adverse experiences are summarized here. And here we have a comparison between the two vaccines. Essentially, the profile looks the same. We have very few vaccine-related serious adverse events in day 1 to 15 following a vaccination. No vaccine-related death, very few discontinuation. Really, the only difference that catch the eye, so to speak, is at injection site, where there is a higher frequency for injection sites. So we will look at the data in more detail. I'd like to say it's not a surprise. We have more adjuvant and we have also more antigen than in the 4-valent vaccine. So let's look at the injection site data. And here we're looking at erythema, pain, and swelling, which are the most, by far the most common. And we can see that there is a difference for all three. And the difference is statistically significant. Due to the very high number of subjects, we can demonstrate that difference. From a clinical standpoint, most injection site adverse events were of mild or moderate intensity with both vaccines. So we anticipate that the clinical significance of this difference will be minimal. Looking now at systemic vaccinated adverse events. So here we have all systemic vaccine-related adverse events with an incidence of more than 2%. I would like to remind you that we are looking at a study of 14,000 subjects and only 5 events make that cut. And all these events are everyday life events. They are commonly used in many vaccine studies, and there is really no substantial difference between the two vaccines. We have observed 4 vaccine-related serious adverse events, 2 in the 9-valent group and 2 in the 4-valent group. None of these required hospitalization except for 1 day for the event of headache in the 4-valent group. These adverse event terms have already been seen previously in the 4-valent group, in the 4-valent clinical program, and all events were promptly resolved. So in conclusion for Protocol 1, in terms of efficacy, we've demonstrated non-inferior immune responses for the original types. We've demonstrated approximately 97% reduction in disease for the additional types. The vaccine appeared well tolerated in more than 7,000 young women, and the adverse experience profile was generally comparable between the two vaccines. We noted a higher frequency of injection site adverse events with the 9-valent vaccine. However, most were of mild or moderate intensity. Let's go now to the protocol 2. Now let's switch gears. And you remember we had 4 goals, and one important goal is to assess immunogenicity in adolescents. Protocol 2 is also an international study to demonstrate non-inferior immunogenicity in adolescents. So we are comparing the young women, population where the vaccine efficacy has been established, to adolescents, and we'd like to demonstrate the same immunogenicity. Obviously, there is also safety here. Study includes adolescent boys and girls, and as well as young women. It's an open-label study. All subjects receive the vaccine as a 3-dose regimen, and we assess immunogenicity at day 1, month 7, safety throughout the study, day 1 through month 12. In terms of antibody titers, you see here the GMT in girls versus young women. The girls are in pink, and the young women are in red. And it's clear that we met the non-inferiority. The GMTs are higher in adolescent girls compared to young women. This is very reminiscent from what has been seen in the 4-valent program, where there was a similar comparison made for the 4 original types. And the GMT ratio were also like here around 2. So once again, we have again another similarity between the 2 vaccines. We've seen similar safety, seen similar efficacy. We see also similar immunogenicity profile. Let's go now to boys. Next slide. And the boys also. I'm sorry. Before we go to boys, let's look at seroconversion rates. And this slide shows you that most subjects become seropositive for 9 types after a 3-dose regimen, both in the girls cohort and the young women cohort. Now we are going to talk about the boys. And as you could have anticipated, the boys are in blue. And so we have boys in blue and young women in red here. And like the comparison between the girls and the young women, the boys have a higher immunogenicity, with also GMT ratios that are around 2, some types even close to 3. And so, you know, we have clearly a superior immunogenicity as well here. And seroconversion rates were very good. Virtually all subjects seroconverted to all 9 types. Safety. This is a safety summary of vaccine-related adverse events, day 1 to 15 following any vaccination, and it's apparent that the safety profile is similar between the 3 demographic groups. Safety might even look slightly more favorable with the boys, which is also similar to what had been seen for the 4-valent vaccine in the 4-valent clinical program. So in conclusion, we've demonstrated non-inferiority of immunogenicity in adolescents compared to adults, which supports bridging the efficacy findings in young women to the adolescent population. And the vaccine was generally well tolerated at all age and genders. And let's finish now with Protocol 9. So Protocol 9 was an immunobridging study, but it asked a different question. It asked, how about the immunogenicity of 4-valent versus 9-valent in adolescents? So let's look at the next slide. That was a smaller study, double-blinded, with 2 cohorts, one administered 9-valent, one administered 4-valent. And we looked at immunogenicity day 1 and month 7, similar design as the other studies. And compliance was very good. Most subjects completed the study, received the 3-dose regimen. And non-inferiority of GMT was demonstrated. Here we are looking at the 2 oncogenic type 16 and 18, and really the immunogenicity is comparable between the 2 vaccines. So it mirrors the data in young women. It adds confidence that the 2 vaccines have the same profile. And the next slide shows type 6 and 11, and similar observation. We have similar immunogenicity for the non-oncogenic genital wart causing types. most subjects seroconvert, actually 100% subjects seroconvert after a 3-dose regimen in both vaccine groups, which shows that the vaccine was highly immunogenic. Next slide. So in conclusion, we've demonstrated comparable immunogenicity with the 2 vaccines also in the younger population. The safety profile was comparable between 9-valent and 4-valent in that group. For the sake of time, I didn't show the data. And here again, most injection site reactions were of mild or moderate intensity with both vaccines. So in conclusion, we believe we have a successful clinical development program. We've met all efficacy and immunogenicity objectives for the key pivotal studies. We've demonstrated non-inferior immunogenicity for the original types compared to the 4-valent vaccine, approximately 97% protection against disease caused by the additional types, and non-inferior immunogenicity in adolescents versus adults. The vaccine was generally well tolerated. These 3 studies taken together include more than 10,000 subjects, and the adverse event profile was generally similar to that of the 4-valent vaccine. There are more data to present, and we'll keep that for the next time. We mentioned concomitant use. We mentioned also assessment in prior Gardasil recipients. And study in young men hopefully is going to be available before the end of the year, and we'd like to show that also promptly to this group. Presently, the 9-valent vaccine is still an investigational product, even though the studies are completed. The product is under now FDA review, and it's under standard regular review. And the FDA has recently accepted the file, validated the file, accepted to review the file, and we expect regulatory action within the next months, the next 3 months. Thank you very much.
Thank you. Let's open for questions. Yes, Dr. Jenkins.
So in terms of adverse events, for the 4-valent vaccine, people have experienced syncope following the vaccination, and I didn't see that as anything that you mentioned in terms of the study.
Yeah, we monitored all systemic adverse events. So the events that were shown are the events that are the most frequent. We monitored syncope with fall. There was no event of syncope with fall during the study. And obviously, we've taken also precautions. So subjects were observed, and there was no event like that. Anecdotally, there was one event of syncope prior to vaccination also.
The thought of it.
Dr. Riley.
Yes, thank you. I was just curious, and you may have mentioned it, and I just glazed over. What does this look like in HIV positive? Have that sub?
No, so the question, HIV positive women have not been studied. All the studies have been done as is common in vaccine study in healthy subjects. There has been a study in HIV positive children, 7 to 12 years of age, for the 4-valent vaccine. This was an academic study. And given that the immunogenicity profiles of the 2 vaccines are very similar and consistent, we would expect the same safety and immunogenicity profile in that population as when it is administered 4-valent.
Quick question on any differences in side effects between the first, second, and third dose in terms of the profile?
Yes, there are differences, and the main difference is in terms of frequency of injection site swelling and injection site erythema. The frequency increases by dose, and it's similar to what has been observed previously with the 4-valent vaccine.
Yeah, and as a follow-up to that, do you have any data on women who may have received 3 doses of HPV-4 with regard to injection site reactions in these women if they receive this HPV-9 vaccine?
So as I mentioned at the beginning of the talk, we have conducted one study. It's Protocol 6, and one of the primary goals was to look at 3+3, and we have found that the safety profile was acceptable, and that there was no major increase in terms of injection site adverse experiences.
Dr. Nuzul.
Thank you. That was really very comprehensive and clear, and really across multiple studies and populations, you're showing this 9-valent vaccine to be comparable to Gardasil, which is great news. There was just a press release today that reported that Gardasil received a positive opinion from the EMA for a 2-dose schedule. So I'm wondering your clinical development plan for the 9-valent 2-dose schedule.
Yes, sure. So as I mentioned at the beginning of the presentation, we are interested and we recognize the importance of this topic. A 2-dose study has been initiated for the 9-valent, and we want obviously first to demonstrate the feasibility and value, and immunobridging is certainly an important part of it. We recognize also that a long-term follow-up in terms of long-term effectiveness, in terms of the duration of protection conferred by 2-dose versus 3-dose, is also a critical question. It's a little bit premature to talk more about the plans at this stage, but we have an immunogenicity study started.
Dr. Sawyer.
As a follow-up to that, how long has your long-term follow-up of the quadrivalent 2-dose program been, and therefore what could we expect for the 9-valent?
So we have, just to make sure I understand the question, we have long-term follow-up for a 3-dose regimen for the 4-valent. That's your question, right?
Oh, I was reacting to Dr. Nuzul's comment that the EMU just approved a 2-dose study.
Yeah,
favorable opinion.
Favorable opinion.
At this time, there is no evaluation of a long-term effectiveness for a 2-dose regimen.
Okay, thanks.
Any additional question? Okay, see none. Dr. Markowitz.
I'm going to do 3 things. I'm going to just give a brief summary of what's been discussed, talk about future workgroup plans, and then go right into the updated ACIP statement that we would like affirmation of today. So just very briefly, today's presentation, we heard 2 presentations on type attribution HPV-associated disease, both the high-grade cervical lesions and the HPV-associated cancers. About 50% of the high-grade lesions are attributable to 1618, about 25% to the 5 additional types in the investigational 9-valent vaccine, and the largest percentage of lesions in all racial ethnic groups are attributable to HPV-1618. There are some differences by race. For HPV-associated cancers, 62% attributable to 1618, a range depending on the type of cancer, and about 11% attributable to the 5 additional types. And again, the largest percent of HPV-associated cancers in all racial ethnic groups attributable to 1618. Some differences for oropharyngeal, but not for the other cancers. We also just heard this very clear summary of some of the data from the 9-valent clinical trials just to highlight that we heard the pivotal efficacy trial among females 16 to 26. There was non-inferior immunogenicity for 611, 1618 compared to the quadrivalent, and about 97% protection against disease related to the 5 additional types. And the immunobridging studies showed non-inferior immunogenicity in adolescents compared to adults. This is an expanded version of the timeline that Dr. Bikini presented. It's our estimated timeline for presentation and for consideration of the vaccine by ACIP. So today we heard the types on attribution, we heard the clinical trial data. In June, we're hoping to hear the data on concomitant administration and the data on the question that was just asked about non-valent vaccine in prior quadrivalent recipients. And we're also planning to hear the health economic data. And then in October, we hope to have the GRADE data. We also were hoping that the data at that time will be available from the immunogenicity study that's currently ongoing in males 16 to 26 years of age. There'll be discussion of options and for recommendations. And then if the vaccine is licensed, there would be a vote in February. So the considerations, a variety of considerations are being undertaken by the work group. One, of course, is for a recommendation of routine vaccination at age 11 or 12. We're also considering the recommendations for vaccination of older females and males who are not vaccinated at the recommended age, and we're discussing the timing for consideration of males 16 to 26 because, as you've heard, we anticipate that the vaccine will not be licensed in this age group at the time of first licensure. We're also considering vaccination of persons partially or fully vaccinated with quadrivalent vaccine. We already had questions and discussion, so I'm going to go on to mention that other HPV vaccine workgroup activities, as you can see, we're currently focusing on the investigational 9-valent vaccine, but we're also doing a variety of other things. We have ongoing review of post-licensure safety data and coverage data, and we're also keeping abreast of the data on reduced dose schedules for HPV vaccination. We'll be reviewing those in more detail in the future. I do want to mention briefly something about the reduced dose schedules because there was a question at the last ACIP meeting. There is global interest in these simplified schedules for HPV vaccine because they would facilitate delivery and reduce some of the logistical challenges. There's a variety of data available on 2-dose schedules, both for the bivalent and the quadrivalent vaccine. These are primarily immunogenicity studies, but there's post-hoc evaluation of data from an efficacy trial for the bivalent vaccine and post-licensure effectiveness data for the quadrivalent vaccine. Some jurisdictions are using 2-dose schedules in their national or provincial immunization programs around the world. The EMEA granted marketing authorization for Cervarix, the bivalent vaccine produced by GSK, as a 2-dose schedule given at 0 and 6 months for girls 9 to 14 years of age at the end of last year. And as you just heard, Merck has submitted an application to the EMEA for a 2-dose schedule as well. And the 9-valent vaccine is currently in immunogenicity trials, which we also just heard. So we'll be keeping abreast of these data and can report back to ACIP. Now I'd like to totally change gears and talk about the updated HPV vaccine ACIP statement. We talked about this at the last ACIP meeting as well. This reviews the evolution of vaccine recommendations in the U.S. The first recommendation was in 2006 for quadrivalent vaccine, which was first licensed in females. The two top orange boxes are the recommendations for females, and on the bottom you can see in the blue boxes for males. The most recent recommendation was made in late 2011 for the routine vaccination of males. Because of the evolving recommendations, after the original ACIP statement that was published in 2007, we published 3 policy notes, which, as you know, are quite short. 2010, there was the recommendation for routine vaccination of females with a bivalent or quadrivalent vaccine, and also for the quadrivalent vaccine, the permissive recommendation in males. And then in 2011 was the recommendation for routine vaccination of males. And this is the only recommendation that was made using GRADE because GRADE had been adopted by ACIP just before that policy note. So the objectives of the updated HPV vaccine statement are to consolidate recommendations for females and males, to consolidate information and recommendations for the bivalent and the quadrivalent vaccines, and to harmonize wording that differed slightly between the policy notes and the statement, and to update background information and data regarding efficacy, safety, immunogenicity, impact monitoring, and et cetera. Now, as you know, this kind of overlaps with our consideration of the 9-valent vaccine, but we feel that the updated statement will facilitate efforts to develop future policy. We have this all in one document. So the committee has seen this document before, and I'm going to just go through, very briefly some particular issues. So the sections that we've included in this statement are very similar to the sections in the 2007 original document. They include background on biology, epidemiology, natural history. We review the clinical sequelae, some of the data on cancers that Dr. Soraya presented today, and the other HPV-related outcomes. We have a section on prevention, treatment, and cervical cancer screening, which has been updated. We have a new section here called Selected Healthcare and Research Laboratory Workers that we've discussed with the committee before. This is on non-vaccine prevention measures. The data on vaccines has been updated with the clinical trial data from the end of study data. We also have some data in here on the cross-protection. We talk about the post-licensure safety data, which was not available at the time of the original statement, of course, in 2007. We have sections on economic burden of HPV disease and cost effectiveness. We have a section on the vaccination program in the U.S. Again, that was not in the original document in 2007. A summary of our rationale, and then the standard sections under the recommendations. This is the main recommendation. In here, we've combined the wording from the male and the female recommendation. When we reviewed this with the committee in October, we had actually a separate statement for the females and males, and we received feedback that we should combine this. So currently, it reads that ACIP recommends routine vaccination at age 11 or 12 years with HPV-4 or HPV-2 for females and with HPV-4 for males. And the vaccination series can be started beginning at age 9 years. We talk about the dosing intervals, and then vaccination is also recommended for females age 13 through 26 years and for males age 13 through 21 years who have not been previously vaccinated or have not completed the 3-dose series. Males age 22 through 26 years may be vaccinated. So this is the combined statement for the males and the females. The other subsections in the recommendation section are the special populations, and the precautions and contraindications. In the document that I distributed to the ACIP members, we had lactating women on the wrong section. It should be in special populations, and it's correct here on the slide. One new section here is history of sexual abuse or assault. Again, we've discussed this with the ACIP several times. And then precautions and contraindications are the same sections we have had in the previous statements. For immunocompromised persons, we are recommending, this is our recommendation, for routine vaccination at age 11 or 12 years with bivalent or quadrivalent vaccine for females and quadrivalent vaccine for males. And it's recommended through age 26 years for immunocompromised persons who have not been vaccinated previously or who have not completed the 3-dose series. And then the recommendation for men who have sex with men through age 26 is also unchanged from our previous recommendation. The new section on history of sexual abuse or assault says, while vaccination will not promote viral clearance or protect against disease progression due to types not already acquired, vaccination would protect against vaccine-preventable types not yet acquired. ACIP recommends vaccination beginning at age 9 years for children and youth with any history of sexual abuse or assault who have not initiated or completed a 3-dose series, and adults through age 26 who are victims of sexual abuse or assault should receive vaccine if they have not already been vaccinated. The precautions and contraindications sections, again, these are basically unchanged. I want to mention that here we say that, of course, HPV-4 is contraindicated for persons with a history of immediate hypersensitivity to yeast, and the pre-filled syringes of HPV-2 have latex in the rubber stopper and should not be used in persons with latex allergy. We removed the statement about single-dose vials because they are no longer available in the U.S. Pregnancy is also unchanged. It's a precaution. We say that vaccines are not recommended for use in pregnant women, and the rest of the wording here is the same wording we had in our previous statements. We have a new section included on monitoring impact on of vaccination, which includes some of the post-licensure impact data. And then finally, an area on ongoing research and future priority areas, activities. And here, as we mention a variety of issues very briefly, including the investigational 9-valent vaccine and reduced dose schedules. We received multiple very helpful comments from ACIP members as well as other reviewers. These were multiple small edits, requests for clarification in some of the background sections, a request for modification of wording of the section on history of sexual abuse, but no change in the recommendation. And we also received requests for including additional data on post-licensure safety evaluations. This is a table that's in the ACIP statement. You've all seen this. I just want to point this out. Here we have listed the post-licensure safety studies that have been done, the population-based studies. This is an update of a table that was included in an MMWR on HPV last summer, and it includes the data from these studies. We added additional text to the ACIP statement to describe briefly each of these studies. And these are just the references from that study, from that table for your information. So in summary, the draft statement updates background information and clinical trial data. It consolidates and clarifies existing recommendations. It does include a variety of new sections, and we have included a substantial amount of post-licensure safety data. This does have to finish going through clearance, editorial clearance here. I'm sure there'll be a lot of additional changes, and we hope to have it published in the spring, however. We do know that there's a lot of new data that's going to be available, and we understand that recommendations will be updated in the future as more data become available. So I think I want to thank the writing team and the workgroup members and the large number of staff that have been involved in all these efforts. And I think we're ready to have questions and a vote.
Thank you, Dr. Markowitz, for a very succinct and thorough report here. I just want to remind that it's our goal at ACIP to renew, revise, or retire statements about every 5 years. So this is very, very timely, considering everything that's gone together for this. The other mention for the rest of the audience here is all the ACIP members have received this document and have reviewed and submitted comments on it previously. But we have a comment here from Dr. Coyne-Beasley.
I was going to make.
Oh, boy, we are just jumping right. So we have Dr. Coyne-Beasley has made a motion to accept the revised recommendation.
You knew that, yes, exactly. Yes. I've had a chance to review the entire document, appreciate all the work and time and energy and effort that's gone in from everyone who has reviewed it and contributed to it. So that is my motion.
Do we have a second? I see several, but we'll give credit to Dr. Rubin.
I second the motion.
Additional comment? I think this is an attestation to the upfront work done by Dr. Markowitz and the rest of the HPV team. This, I think, for all of us was a very readable. Anytime we receive anything 50 pages to review, is daunting, but this actually read very quickly and went very nicely. So I guess we can go for a vote. We will start with Ms. Pellegrini and go to your right.
Pellegrini, yes.
Rubin, yes.
Harrison, yes.
Vasquez, yes.
Coyne-Beasley, yes.
Harriman, yes.
Kemp, yes.
Bocchini, yes.
Reingold, yes.
Kaplan-Salkout, yes.
Karen, yes.
Uchin, yes.
Tempte, yes.
Bennett, yes.
And so the motion is approved unanimously. We thank again the HPV work group for this. Before you step down, Lori, I'm just wondering if there is any comment or question for the work group regarding kind of the next steps. I'm just reflecting on the discussion yesterday about how difficult it is to consider making a change in an already established and very successful vaccine program, trying to reduce doses. So I'm curious as to what type of information we're going to see on, potential reduced dose schedules in the next coming months.
Well, we've been watching very closely the discussions for the pneumococcal vaccine, but right now we don't have anything planned in terms of bringing anything forward to ACIP. We're just keeping abreast of the data and reviewing what's happening, and we are aware that there's a lot of interest in this area. So I just wanted to make that point, and that's why I raised it, because there is a lot of information. There will be more information coming in. We're happy to present those data, but we're not really ready to consider at this time any type of a motion or a consideration by ACIP.
Dr. Tan.
Thank you, Dr. Samti. LJ Tan from the Immunization Action Coalition. Thanks to the ACIP for consolidating these recommendations. As we all know, having a nice consolidated guidance out there would be really, really useful for the providers to strongly recommend this vaccine. And I want to take this opportunity, in line with what you've just voted on, to point out to you a dear colleague letter that's now in front of you, along with a press release to market the letter, essentially urging providers to not just give a recommendation for HPV vaccination, but the recommendation actually has to be a very strong recommendation. So the tagline for this dear colleague letter essentially is, what you say matters, but how you say it matters even more. And it has been a delight to collaborate with the American Academy of Pediatrics, the American Academy of Family Physicians, the American College of Obstetricians and Gynecologists, the American College of Physicians, and with the CDC to create this dear colleague letter that you now see in front of you. This dear colleague letter was launched 2 days after the President's Cancer Panel, and we are indeed fortunate to be able to collaborate with the Cancer Panel to actually market this letter with their report. And this letter has been very favorably received in the media, and so I just wanted to draw your attention to that. And if there are any questions, you can direct them to me. Thank you very much.
Thank you. Dr. Smith.
Yes, this is Jean Smith, Medical Officer with ACIP. In the last 2 days, we've heard mention of 2 anniversaries, the 50-year anniversary of ACIP, and then Ann Schuckett mentioned the 20-year anniversary of the implementation of VFC. I'd like to inform the audience that this is the 10-year anniversary of Lori Markowitz serving as the workgroup lead. She has worked with several ACIP members serving as workgroup chair, and I can personally say that I'm astonished at her fortitude as problems and issues are thrown up again and again, and she always marches forward with absolute calm. Thank you, Lori.
That was a nice way to end a session.